not the presenter mode. So thanks, Richard and Zubair, for the invitation to talk a little bit about what we do in my lab. I think this is going to be definitely a change of pace from what Anne has just presented, and probably a change of pace in general, because I'm a synthetic chemist by training. And in my lab now, we do polymer chemistry. And overall, we try to develop new materials for biomedical applications. We have a lot of interest in antimicrobial polymers and also in targeted delivery, which will be of relevance to this forum, right? But what I want to talk today is a process we've developed to do kind of in situ or rapid screening of polymers for biological applications, okay? And what we're trying to address here is that unlike small molecules, if you want to understand some sort of a structure activity relationship, and if you see that, molecule 2 has a slightly better activity, you can infer that maybe it is because, molecule 2 has a certain functional group, right? But in polymers, this gets confounded by the fact that polymers may or may not have different lengths. And to give you an early example, this is work by Bob Langer and Daniel Anderson in the early 2000s, where, for example, if you compare these two polymers, and I hope you can see, the only difference between these two polymers is that the yellow one has this cross-linker that has an extra methyl, okay? And you could argue that, oh, the activity is related to that, let's explore that space. But as you can see, that space doesn't really work that well, right? And when you look at the details in the experimental section, you find that actually that is more because the polymer is significantly bigger, okay? And that's what gives the activity to this particular material, okay? So how can we address this? Well, one way of addressing this is what we call post-polymerization modification, where you will make a reactive polymer. And if you choose the right chemistry, then you should be able to change the chemistry on that polymer and prepare libraries of compounds that should have roughly the same degree of polymerization and roughly the same number of units. So hopefully now you can infer that if this polymer works better, maybe it is because I used 3 yellow functional groups, okay? Now this still leaves you with a big problem, which I think is inherent to any material discovery. It is inherent to drug discovery for sure, right? Which is most of these polymers are not gonna work. So basically, you're investing a lot of effort in isolating and purifying materials that are actually of little use for you, right? So we thought it will be nice if we can take this a step further, right? And do the evaluation of the biologicals in situ. If we can find ways of doing the post-polymerization chemistry in an aqueous environment, we should in principle be able to test those materials without having to isolate them. Right? And that would allow us to screen the chemical space that we identify as useful very quickly, like I say, saving us a lot of time in isolation and purification. And obviously this strategy relies on these modulators not having an effect because you may have some excess of those, okay? Now what you need to do then is look at, what we tend to use for post-polymerization modifications, which are what we call click chemistries or highly efficient chemistries, which are chemistries that basically, are high yielding and tend to give very few side reactions or byproducts, and some of them will be, innocuous like water. But if you look at the common chemistries used for these at the moment, like, the famous alkyne azide cycloaddition, very often you rely on materials that are not soluble in water, okay? Sometimes what you will see is that you have a lot of side reactions because either the molecules are not stable, or you get, for example, for thiols, you get oxidation of those materials, right? So what we thought is actually we could use quite a bit of old chemistry, which is the condensation of an amine with an aldehyde. And in the context of this meeting, that's actually the chemistry that is behind vision. So, we know that retinal binds to the protein, to opsin via this chemistry. And actually this imine chemistry of CIF-based chemistry has been heavily exploited in drug delivery, okay? And what is very nice about this chemistry is you can have access to a range of amines with different properties. They're all very water soluble. You have to be a little bit careful because, a simple amine has a pK of around 10, which makes it cationic under neutral conditions or under most biological conditions. And that can have issues with toxicity. I'll come back to that. But some chemistries like alkoxyamines or hydroxides are actually neutral in most biological conditions. And this chemistry is reversible, and that's why it's been used heavily for drug delivery. But if you get the chemistry right, and again, hydroxides and alkoxyamines tend to be quite stable, you can use them in biological applications. Seeing how there was some precedence in making, libraries of glycopolymers, we decided to adapt this methodology and, build on, making polyhydroxides to develop this in situ post-polymerization modification. And actually, aldehydes, you can buy almost any aldehyde known to man from Aldrich, so we should be able to explore a very broad chemical space. Now, I won't bore you a lot of details with the chemistry. We tend to use free radical polymerization in my lab, and that gives us access to what I'm going to call the first generation of these materials. They are easy to characterize by GPC and also by NMR. And actually, NMR is very convenient to monitor the functionalization. Now, in most conditions, we will use a little bit of organic solvent, and that's because very often we like to use hydrophobic aldehydes. I'll come back to this in a minute. And in these conditions, we tend to get around 70% loading if we only use one equivalent of the aldehyde. Now, you can push this to almost full conversion if you use a slightly excess of the aldehyde, and as long as this aldehyde does not interfere with your assay, with your biological application, you're good to go. The chemistry is quite versatile. You can do it in purely aqueous conditions, and most of the literature say it uses lightly acidic conditions. And you will see that, you get, again, roughly good conversions, but often what you will see is there are some functional groups that are unable to be functionalized because they're basically acid and insoluble in those conditions. You can do the chemistry under basic conditions. You can do also the chemistry under neutral conditions. And that allows you to put some of the stuff that didn't work in the first place. And actually, you can use almost purely organic conditions. And then that basically, like I say, opens the Aldrich catalogs, and we can put every functional group known to man. Okay. So what do we use this for? So basically, when we started working on this, we decided that we were going to focus on nucleic acid delivery. And as you've heard yesterday, this is a pretty big challenge in biology. You're basically trying to deliver a very large molecule like a nucleic acid that is negatively charged and actually it has a very high charge density, right? And at some point, it will have to cross a biological membrane that is hydrophobic and is actually mainly negatively charged as well. So that's basically a no-no. That's not going to happen. And what nature does, and we do, is we tend to complex these negatively charged materials with positively charged materials to give what can be called, a polyplex, a lipoplex, etc. And that is then able to cross across membranes. There's a range of materials you can use. Lipids are quite timely because they are the core of some of the messenger RNA vaccines that we're using at the moment. But you can also use nanoparticles, polymer. And being a polymer chemist, we focus on developing new polymers. And as I said before, one of the challenges with positively charged polymers is that they tend to be toxic. And if you look at one of the most common polymers used, polyethylenimine, it is quite toxic. Now, how does nature address this? There's mainly two ways. First of all, it tends to deliver or use these kind of materials that complex DNA at the site of action. So it's very local delivery, and then eventually gets degraded. But from a chemistry perspective, what is fascinating is that it uses a range of amino acids. Some of them are charged and responsible to complexing the nucleic acid. Some of them will probably buffer the pH a little bit, and some of them are hydrophobic. And it's this balance between these amino acids and the shape of the peptide that actually does the job. As a synthetic chemistry, making a polymer with all these monomers is very challenging, right? It's very difficult to optimize. But we can use this chemistry, we can get aldehydes that actually mimic all of these amino acids. So in principle, we should be able to make polymers that carry all these functionalities. So far, we haven't explored such a complex polymer composition. We're mainly working with binary compositions where we explore, a cationic component, and a hydrophobic component. And actually, again, the chemistry is quite simple. Like I say, we tend to use a little bit of organic solvent to dissolve those hydrophobic aldehydes. But depending on the assay, this is not a big problem. You know, it often gets diluted down when you add your nucleic acid of interest, and this is siRNA. And sometimes you will even get further diluted when you put it into your cell assay, right? So, you know, at the end, you may get less than 1%. And I think in this assay in particular, we were using 0.01% of DSO. We have now ways of using other solvents that can be removed, right? So they are not present when we use them. Sometimes what you will see is that the system aggregates because you're using very hydrophobic components, and the magic number tends to be around 50%. There are tricks around this. You can use large hydrophobic aldehydes, which form fast precipitates. Eventually, you know, if you formulate them properly, you're good to go. As chemists, we tend to try to find very simple assays that tell us what we're doing, make sense or not. And one of the assays we do, it's this lipid liposome assay, which is a fluorescent assay that basically tells you whether things are crossing through the membrane or breaking the membranes apart. And that allows you to discard some formulations that are actually very, or are surfactant type formulations, but also make a short screening of, you know, trying to identify what functional groups are useful, right? And basically here, this is an EC50 assay. So the lower concentration, the better, and the higher the intensity of the dye release, the better. And you will see that aromatics tend to be really good. And, you know, aliphatic compounds maybe a little bit all right. And all these heteroatoms and, you know, kind of aliphatic aldehydes don't really work. Now we decided to test this in HeLa, which may be the most boring model, but, you know, for us works really well. And what we find is actually aromatics don't work that well. And basically this shows the difference between, you know, a simple liposome and a cell membrane, right? But what I want to highlight with this slide is, again, our methodology is very modular, and we can very quickly optimize the composition, right? So this is the ratio between the hydrophobic and the hydrophilic compound. And again, without isolating any of these materials, we can see that, you know, 15% of the hydrophobic is the way to go. We can also optimize how much siRNA we release in one single assay. And again, you know, we found that 12 micromolar was the way to go. And basically, this material is not toxic at the concentration we've evaluated, and can deliver, you know, siRNA with the same efficiency of lipofectamine, siRNA max or RNA max, but we're just using 10 times less material. We also use it to deliver other nucleic acids, such as plasmid DNA. And what I want to highlight here is, again, the methodology is very modular. So in the same assay, we can optimize the length of the polymer and the composition of those polymers. And we can see that for plasmid DNA, we basically need a larger polymer, right? The small polymer we use for siRNA doesn't work, and you have to use a polymer that has now 180 units. And actually, in this case, we need to use very long aliphatic aldehydes. And again, we were able to release, to transfect or deliver, you know, significant amounts or similar amounts to lipofectamine. We've also explored using this to deliver messenger RNA, which is very sensitive, and it basically breaks up on looking at it. And it kind of works, it does work. And again, these long fatty aldehydes seem to do the work. So how is this relevant for Orbital? Can we do something with ocular diseases? And yes, we can. We have started working with Rachel, and it's a project where Zubair and Anna are also involved. And basically, we're looking at, you know, can we deliver some of these materials across an ocular model? We now have 2 generations of these polymers, and both of them can cross this model, can go across this ocular model. And actually, generation 2 is, you know, 4 times better. As before, we can explore using different aldehydes to, you know, affect how well this material goes across our model. And we have identified 3 compositions that show promise. But what is actually more exciting is that we can use this to deliver nucleic acids. Like I say, they're really terrible at crossing across the membrane. And we have identified now 5 formulations that are really good at doing that. And we can also use simple drugs, which are a little bit better at going across our model than nucleic acids because they're slightly hydrophobic. But in the presence of a polymer, we get up to a 10 times increase in how much they go through our model. And this is significantly bigger than what you tend to do, which is basically make the drug a little bit more water soluble. And with that, I'm going to stop. I hope I have convinced you that polyhydrazides are very good to screen biological polymers for biological applications. We can do this in aqueous conditions, and we don't need to isolate and purify our materials, which saves us a lot of time. As far as we are aware, the polymers are non-toxics in the model we have used, which is great. And we use this to deliver nucleic acid, and we have a patent on that. And the idea here is, can we use it to treat ocular diseases? So I just need to thank the people that do the job. The ocular work is mainly Tom, but the polymer was developed first by Daniel and then by Oli. Obviously, we can't do any science without collaborators, but also, with the funding. And thank you for your attention. I'm more than happy to take questions.
Thanks, Paco. Fantastic talk. I'll kick off with a question. What key features do you think in, not necessarily even polymer design, but molecular design, give you the ability to transport other molecules?
Okay, so I'll go back to one of the slides, actually, if I can. Can I share again?
Yes, you can. Go for it.
So often people will say that what you need, and this is what nature does to make cell-penetrating peptides, which again are peptides that go in or through membranes and help, for example, viruses infect, right? And again, if you look at histones, they'll have, a high density of guanidinium groups, which are, permanently charged and coordinate to negatively charged RNA. And it's this balance between, positive charge to associate to your negatively charged membrane and hydrophobicity that allows them to go through the membrane. Now, there's a lot of debate whether, again, the shape of the peptide has an influence, right? And you'll see that a lot of these cell-penetrating peptides, antimicrobial peptides, and even, the things that are complex nucleic acids tend to be helical, but some of them are not. And what is very fascinating from a chemical perspective is some of them actually adopt this shape upon interaction. So they're kind of stimuli responsive, right? They will be randomly shaped, which probably minimizes the toxicity. And then when they see a membrane, they adopt the structure they need to go through, much like a screw. I hope I answered your question.
Yeah, thanks. Larry, is that a legacy hand, or you're asking something?
It's a legacy hand, but it's a very interesting talk. I did my PhD in polymer chemistry, and I kind of miss it. So it's in some cases. I was wondering, I mean, what do you find the biggest challenge in terms of functionalizing the polymers? And I know that, we looked at this. We're trying to functionalize polymethacrylic acid with a cyclodextrin at one stage, and it would work one time and then it wouldn't work the next time. How do you find repeatability? I suppose is one of the.
That's a huge challenge actually in polymer chemistry, and listening to Anne's talk, you kind of wonder that there's a lot of lessons there, because again, reproducibility, it's built on how good your record keeping is, so then you can do exactly the same, right? Because if you don't do exactly the same and you add a little bit more of initiator, then it will be a little bit shorter, right? And if you didn't degas as well, then, for example, for radical polymerization, then it may be a little bit shorter. The scaling up here is a big issue as well, because again, 5-minute degassing for a 1 milliliter solution is probably more than enough, but how do you translate that to a 20 milliliter, 100 milliliters, etc., right? So that's a huge thing. And again, I would love to talk to Anne in more detail about this stuff, and how do we get members of the team to kind of do this. Am I still connected?
Yes, you are.
And that was as you were talking, I was thinking, how does this tie in with what Anne was talking about, that translational piece, the reproducibility, because like you say, a fraction of a percent, weight percent more of initiator, and you've got a different chain.
Exactly. Exactly. And it's the same with the aldehyde functionalization. One thing I want to mention with cyclodextrins, we find in our chemistry size matters, right? And if you put a small aldehyde, then you get 85%, right? And if you put a bulky aldehyde like indole, for example, which will mimic tryptophan, right, then it lowers to 60%, right? When you're putting cyclodextrin, which is a chunk, right, it is a challenge, and you have to be careful. Now then it's choosing the right chemistry, and we can talk about the right chemistry if you want.
Paco, is there a limit to what size nucleic acid you can link to your polyhydrogen lines?
So it depends on the polymers. Again, we've been able to deliver plasmid DNA, which is kilobase length. So it seems to work with some of our generation 2 polymer. It seems to work worse for plasmid DNA. So again, it's going to depend on the flexibility of your polymer versus the flexibility of the DNA. Sometimes people argue that one of the challenges with small DNA is that it's too rigid, right? So it's the other way around. Size is not an issue, but rigidity is an issue in compacting that DNA or our RNA in this case into a particle, right?
Thanks, Paco. I think that's fantastic talk. I think it's given people an overview of some things perhaps they haven't thought about at the chemistry level. Zubair, I think we've got 5 minutes. So if you want to give people a quick break to get a coffee.